I start, I want to thank the organization, especially Dr. Dipti Jain, for inviting me to share my work on microRNA. As we have heard for last 1.5 years, that hydroxyurea is the drug with some of the drawbacks for the sickle cell disease patient. And as we know, it ameliorates the disease severity by increasing the HbF. So here I am presenting the role of few microRNAs in hydroxyurea-mediated HbF induction. As we know that erythroid-specific gene at beta-globin gene locus is developmentally regulated by complex series of epigenetic and molecular process. And the proposed epigenetic mechanism, as you know, of HbF regulation includes methylation for reactivation of HbF. As we know that hydroxyurea is a ribonucleotide reductase inhibitor. Initially, it was developed as an anti-neoplastic agent, and later on, it was found to be beneficial for treatment of sickle cell anemia. As I said earlier, its benefit is primarily due to the ability to increase HbF. As we know that hydroxyurea decreases frequency of painful crisis episode of acute chest syndrome, and in some studies have shown that it also reduces the mortality. The preliminary study done in our institute to look for the effect of hydroxyurea on sickle cell patient, clinical condition of sickle cell patient, have shown significant improvement in 98% of the sickle cell anemia. Most of the patients were off transfusion. So, however, the HbF induction by hydroxyurea is highly variable among the patient. The mechanism is exactly not known. And although the direct transcriptional effect or the cell cycle kinetics has already been told, have been proposed mechanism. Lot of studies nowadays has been carried out on microRNA. And microRNA are nothing but molecular regulators that modulate the expression of protein coding genes by inhibiting mRNA translation. Lot of studies have been carried out to see the role of microRNA expression in blood cell development, but hardly any studies have looked for importance of this in HbF induction. There are few studies who have shown the role in sickle cell anemia patient. These are mainly through the literature. Some of what I got is these are the microRNAs who are involved in HbF induction or gamma globin gene increase in the gamma globin gene expression. So main aim of my study was to find out the role of microRNA in hydroxyurea mediated HbF induction. So to start with, we had about 10 patients with sickle cell disease. It was very difficult to get the sickle cell patient at baseline. So we only got the 30 patients, mainly at the baseline, 30 normal individuals. Then for follow-up of 3 months after hydroxyurea, we have 20 patients, and for 6-month follow-up, we have got 10 patients. So this slide shows the induction of HbF level by hydroxyurea. As you can see, 20 patients we have studied both at baseline and 3 months after hydroxyurea. That the sample showed, the sickle cell patient showed a significant increase in HbF level after hydroxyurea. This is a baseline. It's 16%, 30%, and 35%. And this graph just showed the individual variation of HbF among the patient. Then we have published some paper on clinical score, and accordingly we tried to see the clinical course of patients before hydroxyurea and after HU therapy. And as you can see that all the patients responded very well to HU therapy, and you can see most of the patients were off transfusion after hydroxyurea. And all the patients showed significant improvement in their clinical condition, which in turn led to a better quality of life. This is just the separation and enrichment of CD71 positive cells. Then we have looked at 10 different microRNAs. This we have selected from looking at the different, we have not have any microarray done. We just selected it from the review of literature that some microRNAs which are involved in HbF induction. The MIR451 was used as a reference gene for this study. Normalization, as you can see, it constantly expressed and remain unchanged in the treatment group. And these other graph showed the amplification plot of different microRNAs which we have used. Here it's showing for the three samples. You can see if you look, this is the MIR210 which has shown the highest expression. This is the control group as baseline, and then after hydroxyurea. Similar result were found for MIR29. Then we also looked at the gamma globin gene expression in these three groups: control, sickle cell anemia, and after hydroxyurea. And as you can see, we observed a fourfold increase in the gamma globin gene expression in sickle cell patient as compared to the control group, whereas after hydroxyurea treatment, we found about eightfold increase. Then we also tried to correlate this gamma globin gene expression with HbF level. And we found a positive correlation between the gamma globin gene expression and HbF level among three groups. This is control at sickle cell patient at baseline and the hydroxyurea. This slide shows the relative quantification of 10 microRNA which we have studied. And here you can see that HbS homozygous patient, which are in pink, showed a significantly higher expression of the 7 microRNA as compared to the control group, which is in blue or purple. And after hydroxyurea, the expression were still increased. Whereas you can see the 3 microRNAs, MIR320, 96, and MIR223. This is the logarithmic scale, showed the downregulation of the these microRNAs, whereas MIR323 showed the overexpression after the hydroxyurea treatment. Then we tried to see the effect of hydroxyurea on expression of these 10 microRNAs. Here you can see, here we compared it with sickle cell homozygous with adult, and then HbSS at baseline and after hydroxyurea treatment. And you can see that 8 microRNAs showed a significant increase in the expression, whereas these 3 microRNAs, MIR494, 210 and 144, showed the highest expression, whereas these are the 2 who have shown the downregulation. This just shows the heat map illustration relative to the change fold in the microRNA expression. This is what I said we have used as an endogenous control, which has constantly shown the expression. Then we tried to correlate the expression of all these 7 microRNAs which showed the higher expression with HbF level and gamma globin gene expression. And what we found is that significant association between the HbF, this particular slide is showing for MIR210. We found a significant association between the HbF and MIR210 expression. Similarly, we found a positive correlation when we compared it with the gamma globin gene expression. This is for other MIR61, MIR29. Then MIR96 showed a decrease in expression was strongly associated with the increase in HbF level, and similar was seen in the gamma globin gene expression. So after this, we just tried to see that how these microRNA might be working through with genes. And this we just tried to, looking at the other literature, we have drawn this cartoon. And what we found is MIR144, which is mainly involved in the human erythropoiesis, work through these genes. Then these 3 overexpression of these 3 microRNAs, it is seen after the hydroxyurea therapy in sickle cell anemia, had shown that it protects cells from hypoxia-mediated apoptosis. Then MIR210, you can say, directly work on gamma globin gene. Then MIR96, which had shown a downregulation in star, is an inhibitor of gamma globin gene mRNA. This just shows the role of what I said of different microRNAs in the induction of gamma globin or HbF. Then as we were having these few microRNAs, we predicted the target genes of a microRNA using different prediction software. And what we found surprisingly is most of the microRNA share a similar target of well-known BCL11A, MYB, and KLF1, which are known to be associated with increased HbF level. So to summarize my study, the 8 microRNAs showed a significant increase in their expression after HU therapy, but only 4 microRNAs showed association with raised HbF, providing their role in HbF induction after HU therapy. Given that these 4 microRNAs play a crucial role in gamma globin gene regulation and induction of HbF, its expression analysis might have diagnostic and therapeutic value, and therefore can be used to at least understand the function of HU-mediated HbF induction. This study is limited to a small cohort of patients, and we still have to validate the target of mRNA, which we are going to do now. However, with their essential role in HbF induction, these microRNAs offer a great potential medicine future, and this finding can be expanded to a mouse model. So basically, these preliminary studies provides an initial evidence for microRNA regulation in HU-mediated HbF induction. Last but not least, any study is always a teamwork. So I take this opportunity to thank all my lab members for their support, and especially my postdoc, Dr. Madhvi Sawant, whose work actually I have presented now. Thank you.